So it's a great question, and so you can't do it prospectively now because it would just take too long. And so what we know is when we introduced Velcade and Revlimid maintenance compared to a single agent maintenance or no maintenance, our 10-year survivor rates went up from just over 30% to over 50%. And so we're not about to go back. The issue is, when is maintenance maintenance and when is it not maintenance? And so there's some implication that for maintenance you need to continue it until the disease progresses. I think my contention now is that 10 years plus for more than 50% of the patients could cause more trouble than it's worth. So we took just a random academic decision to say we're going to continue maintenance for 3 years and stop. And so if we stop at 3 years, I think we get very good long-term outcomes. So maintenance for us is 3 years, but there's still a raging debate about it.
Yeah, as Gareth has mentioned, it is a debate. So we continue maintenance until progression. That's as it is in 2017, because the data with all the maintenance studies is to progression. There are others, you guys are doing 3 years, the French are doing even less in terms of maintenance. I think what we don't know today is, what we do know is maintenance is a good idea. What we don't know is when can you stop maintenance, or what is the duration of maintenance. So this 2 years, 3 years until progression are random, kind of who feels comfortable with what. But this is something we are absolutely going to be addressing, and this is where those tests that we talk about become useful. And the question then is, at the 2-year time point, if you're MRD negative, or the 3-year time point, if you're MRD negative, can I stop my maintenance and will I be okay? Do we have the data to speak to that just as yet? We don't. And those are the kinds of studies wherein we will get that information now 5 or 6 years down the road. So again, these are open questions. I don't think we have answers to any of them. The other thing I will also add is, as of right now, we are saying maintenance is the way to go. I did show you that we are talking about now even combining 4 drugs. So there's one thing in terms of toxicity that we give you with the drugs that you're on, in terms of physical side effects. There's also this whole notion of financial toxicity. It's not easy staying on these drugs indefinitely. And specifically, if we start thinking about combining 4 and 5 drugs together. Now, is it possible that when you combine all of these 4 drugs, you're going to get such deep and durable responses that you will not need maintenance? Those are also kinds of the questions that we will be asking. So yes, as of right now, maintenance, the answer to how long, we don't know, and hopefully we'll address some of these.
So just to add to the confusion, I guess, when everybody talks about maintenance, if you actually go back and look at the clinical trials, not many of the patients stayed on the drugs until progression, okay? Essentially what happens is, with time, patients find that they develop little niggly side effects. So usually what happens is when you first start a drug, if you're going to get any side effects, you get acute bad ones. But when you've been on the drug for some time, you begin to get little ones which, maybe if it's just one you can cope with, I don't know, the bowels, for instance. But then maybe you start getting the bowels and tiredness. And after a while, around about the 2 to 3 year mark, you find that patients have got a bunch of side effects from their maintenance. And so many patients will actually come off there through patient choice, rather than through any kind of other kind of thing. Because, as Noopur said, if we're going to continue it on to disease progression and patients are not progressing for 10 years and you've got awful niggly side effects for more than half of that, that's not quite what we're intending for as well. So I think there's 3 things in here. There's the actual data Okay, there's the biological and the clinical side of things, and then there's the patient and the side effect side of things, and all of that needs to get put together.
So it's easy. So if you have a 50% reduction in your M spike, you're in a response, and that's called a partial response. So that can be a lot of disease in your bone marrow. When there's no detectable M spike, no detectable light chains, and there's less than 5% plasma cells in the bone marrow, that's a complete clinical response. When you use a molecular test, and that's negative, that's called a stringent complete response. So the vast majority of our long-term survivors are MRD negative, so I suspect you're in a stringent complete response. No, it's just like difficult to design, because when the outcome is good, and people in those settings really do have good outcomes, and so the average survival could be 7 years. To see a significant clinical difference, you have to have 3,000 people in the trial. And so that's why you have to develop these surrogate or intermediate endpoints, because we're a victim of our own success. People are doing really well, but then it's very difficult to get new treatments into that group without a change in the way we set up the clinical trials. So there aren't really studies in that setting. We're putting a group of people into a maintenance study, but that's slightly different.
There is a vaccine study, but that's more kind of post-transplant. It is not for people who've been in remission for a long time. So David Avigan from Harvard is doing the study, and what he's done is used a fusion vaccine. You have to take your own myeloma cells, fuse it with dendritic cells, which are immune cells, and it's actually going to be happening nationally. But you have to be doing it after a transplant. The idea of using a vaccination to kind of keep that remission going. But there's nothing for people like what you're describing, where the protein is going up a little bit, and can we go in and do something about it? I don't know if that's going to be a strategy for you just now, because to be able to do that DC fusion vaccine, they should have harvested your bone marrow at the time you were diagnosed, and we needed to have a few of your tumor cells. Right now, you're in a really good place.
No, that's
so
right.
Vaccine cannot really be made for you. As far as consolidation is concerned, consolidation is a weird kind of a thing that we've come up with now. And it was more kind of some of the European studies that ended up using this whole word consolidation. In the old days, to us in the myeloma world, consolidation was the transplant. You had your initial therapy, you used the transplant as something to consolidate whatever response you got from your initial therapy, and then you were maintained on something. This notion of consolidation after a transplant has come up more largely because in Europe, the way drugs were approved, they really did not have Revlimid up front. So they were using Cytoxan-based regimens there. And they wanted to use some kind of an IMiD at the back end to make sure that everybody was in an equal playing field, because people were getting all kinds of responses, or all kinds of chemotherapy up front. The only other thing I will add is when we are collecting stem cells, we collect after 4 to 6 cycles of whatever your initial treatment is. And some of us feel that that is not enough of the proteasome inhibitor therapy, and therefore doing a couple more cycles at the back end where we give you that proteasome inhibitor may be useful. As far as using 1 or 2 drugs, I'm going to let Faith address that.
I think there's lots of discussion and debate about how many drugs you should have in the maintenance phase. Okay, you're right. Some places will just use a proteasome inhibitor, such as Velcade, or the new oral one, which is being used, ixazomib. Others will just use Revlimid. There's also clinical trials at the moment looking at the antibody Darzalex in that setting as well. And then others will do the combination of the drugs. Nobody truly knows the answer, okay? I think, as I say, we've ended up on the 3 drugs, but I think the key is to at least try and have some therapy, the idea being that, the way I usually describe it, if one of the myeloma cells suddenly wakes up, there's something there to give it a bash back down again. Okay, so, and I think, as we said before, making sure it's tolerable is also important.
And I'm
Gareth's going to disagree.
Oh, I'm sorry.
No, I was going to say there are sometimes you should be happy about the position you're in, because you seem to be in a very enviable and good position. So carrying on as you're going is, you should be very reassured. So I'm not very good at that, but I think it's clear that the CAR-Ts, the bispecific antibodies, the imaging technologies are going to be so much better that we'll be talking about cure for definite and in a large proportion, and we'll definitely have segmented people, and the very high-risk patients will be known up front and be treated differently. I think that's where I think it's all going.
I think 5 years from now, you know, I do think we're going to be using the word cure very boldly. I think we're already curing a fraction of myeloma patients. It's just not high enough. But 5 years down the line, with everything we have in terms of technology and the things which are in development, we are all so encouraged by what we've seen in very late stages of disease. And the way things work is all of this is going to come up more upfront. So we're going to be using them more early on in the course of the disease. And the hope is going to be that we're talking about continuous therapy and myeloma maintenance and so on and so forth. Those are not questions we're going to be asking, because with drug treatments like the CAR T cells, it's a one-off treatment where you're not on any treatment after that. And I do think we're going to see that going forward in the future.
So I think what will happen in 5 years' time is that the patient will come into clinic after their 3-day testing the Arkansas patients know about. But whereas at the moment when we do that testing, we know where the myeloma is from the imaging, and we know a little bit about the genetics, I think we'll know all about the genetics and all about the immune system. And at that point, we'll then be able to put the patient's story and how they are with their immune profile, their genetic profile, and their radiology profile, and we'll be able to say, right, this is the best treatment for you now. And I hope and I pray that's where we're at.